Hi everyone, here's the video that we've prepared for you to revise for the exam in terms of the histology content that you've had delivered this year, or to introduce some of the new concepts that will also be examinable. This is a reminder that all content presented in this video is examinable. You will not be examined on anything that you haven't seen in this video in terms of the histology for this semester.
The tissues that we'll be looking at are the kidneys, pancreas, gastrointestinal tract, bone, ovaries, neurons, and blood.
In terms of the functions of the urinary or the renal system, there are three main things that generally the kidneys are responsible for. So there's the filtration of blood plasma, then reabsorption of a lot of water and glucose nutrients back into the blood, and then the excretion of the filtrate, which is termed urine. So with looking at this cross-section of a kidney, what are sort of the three main segments that you'd separate this into?
So I think it would start with the renal cortex, and that would be the outermost or the cortical layer, as most of you would be familiar with other terms that begin with cortex. Anything that means cortices just means on the outermost. And then we've got the medulla, which is in the middle, and the way that I remember that was medulla starts with M, so it sounds like middle. And finally, we've got the innermost region that's known as the renal pelvis.
All right, great. Thank you, Vith. So I think that before we move on to the next slide, I'll just like to remind you that the cortex area is heavily vascularized. So this is where most of the filtration and most of the reabsorption will occur. Moving closer or zooming into the kidney itself, let's look at the nephron and how it spans across the regions that we just chatted about. So Vith, can you tell me what we're looking at over here?
So the nephron is the functional unit of the kidney, and it spans from the cortex all the way down to the renal medulla as well.
So in particular, let's go through some of the structures, just basic structures of the nephron before we move into the next slide.
So we start with the renal corpuscle in the cortex. The renal corpuscle is made up of a ball of capillaries or a tuft of capillaries called the glomerulus, and it's surrounded by the Bowman's capsule. And then from there we lead into the renal tubules. First part of the renal tubules are, Daniela?
That will be the proximal convoluted tubule, so the PCT.
Excellent.
Then follows into the loop of Henle.
And can I just mention, the loop of Henle dips down into the medullary region, or the renal medulla.
Excellent, great point. And then we continue on to what?
The DCT, or the distal convoluted tubule. So it's called distal convoluted tubule because it's a tube, it's convoluted, meaning it's windy, and it's distal because it's furthest away from the renal corpuscle, as opposed to the PCT, which is proximal to the renal corpuscle.
Excellent. So from the DCT, the filtrate will drain into what?
So from the DCT, the filtrate drains into what's known as the collecting ducts, and collecting ducts just serve as channels to eventually drain the filtrate into what's known as the ureters. The ureters lead into the bladder where the urine is stored, and finally when we're ready to excrete it, we'll do that through the urethra.
Let's look at the structures that are involved in filtration of plasma, reabsorption and excretion of urine more closely. So let's start over here with, let me get the right tool on, with the renal corpuscle. So Vicky, can you tell us a bit about what's involved in this structure?
So all three of those processes, filtration, reabsorption and secretion, require blood supply. So the way that we get blood supply into the renal corpuscle is through the afferent arteriole, so that's AF, and the way that blood supply leaves the filtration unit is through the efferent arteriole, EF. Now the afferent and the efferent arteriole, in the middle of both of them you will find the glomerulus. So the glomerulus is also known as a tuft or ball of capillaries, and we just want a large surface area of capillaries in there so that we can maximize filtration. Surrounding the glomerulus we have what's known as the Bowman's capsule, or the glomerular capsule.
Following on from the glomerulus, once the filtrate enters the capsule, the first part of the nephron is going to reach is going to be the proximal convoluted tubule, known as the PCT. What happens next, Vithia?
So from the PCT, the filtrate then flows down into what's known as the loop of Henle, or also the nephron loop, and that's shown here. From the nephron loop, we then the filtrate makes its way back up into what's known as the distal convoluted tubule, or the DCT. Finally, it then enters the collecting duct and eventually gets into the ureters.
Excellent, thanks. Would you mind discussing the function of some of the structures of the nephron?
So the PCT is mostly known for its reabsorptive ability. So it's going to reabsorb lots of water, glucose, and any of the real important nutrients that we need. The loop of Henle, on the other hand, is really important to create a concentration gradient. This is also known as the medullary concentration gradient because the loop of Henle, or the nephron loop, is found within the medulla. So this concentration gradient is really, really important because it creates passive reabsorption of water from the tubular lumen, or the tubular fluid. What that means is that we're trying to resorb as much water as we can from the tube back into our blood supply because we want to keep water, or we want to save as much water as we can.
So the water gets reabsorbed mainly down the descending arm of the loop of Henle, and the solutes get reabsorbed back into blood mainly on the ascending arm of the loop of Henle.
So following that, in the DCT, we're just going to make some last minute changes that's needed based on our homeostatic state and our metabolite state. So that's where you'll get some reabsorption and also secretion happening.
Other than filtration of blood plasma, reabsorption of water and nutrients, and excretion of waste, the urinary system, I guess the kidneys, are really important in regulation of blood pressure and monitoring of that. How does that occur, Vithia?
So the nephron essentially needs a way of detecting blood pressure and to be able to figure out how much filtration is happening so that we can regulate that accordingly. And the feature that does this is collectively called the Juxtaglomerular apparatus. It sounds like a big word, but it essentially just is an apparatus or a mechanism that's near the glomerulus, or juxtaglomerulus. Specifically, it's the macular denser cells that are really important for this because they are closely packed nuclei that sit within the epithelium of the distal convoluted tubule that we can see here highlighted in green.
Looking at the endocrine system here, so Vithia, as soon as I look at these slides, it'll take me probably about half a second before I realize that I'm looking at the pancreas. Can you please clarify what the distinguishing features of pancreas in a histological section are?
So the pancreas is both an endocrine and an exocrine function, so therefore they've got two distinct populations of cells. Here we can see all around, or sparsely distributed in the section, pancreatic acinar cells, and these serve the exocrine function of the pancreas. That is to secrete digestive enzymes and juices to serve as an accessory gland for the digestive system. But more importantly, we can see little clusters of cells that serve the endocrine function of the pancreas, here and here, and these are collectively called islets of Langerhans.
Looking at the islets more closely, can you please tell me what cells make up the endocrine portion of the pancreas?
So the islets of Langerhans are essentially made up of alpha, beta, delta, and F cells. What's important for you guys to know though is that you can't differentiate these cells in basic H and E sections. We need to be able to detect or immunohistochemically detect certain hormones that are produced by these cells individually to see which cells they are specifically.
Could you please tell me what these cells collectively do? What's their function?
So collectively, like I said, they serve an endocrine purpose. So we're expecting them to produce the main hormones that we need, and those hormones are insulin, glucagon, and somatostatin.
When looking at the gastrointestinal tract, what we want you to be able to do at this year level and in more detail in your coming year levels is identify, just by looking at the identifying features, maybe anatomically or histologically, identify the segment of the tract that you're looking at. So let's identify some of those key features that are present throughout the gastrointestinal tract. So initially looking at the image on the left, we're starting from the lumen outwards. So we've got the mucosa. Just underneath the mucosa, we've got the submucosa. Following on from that, and here you can see the two different layers, we've got the muscle layer, or the muscularis externa. And then the most outer layer of the tract, we've got the serosa, and in some parts of the GIT, it's also termed adventitia. So in this section here, we can see that we're probably in the small intestine simply due to the villi forming into the lumen. But let's not focus too much on that. What I want you to understand are the layers that are consistent throughout the gastrointestinal tract, irrespective of how they may structurally change slightly as we move through. So let's move into the image on the right. And the gastrointestinal tract will always be covered with an epithelial layer. Underlining the epithelial layer, we will always have a lamina propria. And then throughout the gastrointestinal tract, it's really important for you to remember that we will have a thin layer of muscle. And now we're not in the muscularis externa, but this layer is called the muscularis mucosa. Underneath the muscularis mucosa, we've got the submucosa, the supportive tissue of the mucosa, and then we will have sometimes 2, sometimes 3 layers of muscle depending on the segment of the gastrointestinal tract that we find ourselves in.
So Daniela, here we have a histological section of the stomach on the left. And what can you tell me about the structures that you see?
All right, great, Beth. So you know it's the stomach because we're seeing these gastric pits that are forming. So you're seeing the epithelium sort of dip into the layer. But again, as I said, we're not really specific at this year level on identifying where we're at. What we really want to see is identify the features that are common to every stage or every area of the gastrointestinal tract. So here, let me just get the right tool on. So here we can see the epithelium, and because we're in the stomach, this layer forms invaginations or gastric pits that sort of fold inwards. As I said on the slide previously, underneath the epithelium we always have the lamina propria, and this thin, really pink layer is the muscularis mucosa. Moving on from there, we've then got the submucosa, the paler layer that isn't as cell rich or as fiber rich, and finally, just before we reach the serosa down here, so let's label that first, the serosa. Finally down here we'll have the thick layer of muscle, so the muscularis externa.
So Daniela, what image do we have on the right then?
Great question, Vidya. So over here on the right, we are looking at the small intestine, and it's really difficult to distinguish that when comparing it to the left because this image is taken at a much higher magnification. But an identifying small intestine, but the identifying feature of this is this border here of microvilli. So there's a whole villus that sits underneath here, and these tall columnar epithelial cells. So we're looking at epithelium here. And Vidya, would you have any idea of what these cells that are sort of clear in appearance and are labelled as 2 sitting amongst the epithelial cells are in the small intestine?
So those cells are goblet cells, and therefore they are mucus-producing cells, and producing mucus so that they can line that region of the gut.
Thank you. Over the next 3 slides, let's have a look at different types of bone that we have in the body, how their constitution differs, and how they differ histologically. So let's start looking at compact bone. So here in this image, or the diagrammatic representation on the left, you can see that most of this is compact bone, and it corresponds to the histological image on the right. So Vidya, would you like to take us through what we're seeing in compact bone?
So a key feature of compact bone are the systems that we call Haversian systems or osteons, and that's what we can see here. Osteons are made up of concentric lamellae, and that's really clear in both the diagrammatic representation on the left, and you can sort of make that out in this histological image on the right as well. With the lamellae and the concentric layers, they're essentially made up of osteocytes that sit within lacunae. And here is an example of one osteocyte that's sitting within the lacunae, and they stick the processes out, which gives it that concentric layering appearance. What's also important is that in the middle of the osteon, we have what's known as the Haversian canal or the central canal. Both of these terms are interchangeable, and you will see them being used in textbooks.
Why do we have this canal, Vidya?
So with the osteon, because it's hard matrix tissue, we need to be able to get some blood supply into these cells, such as the osteocytes. And the way that we get the blood supply in is through this canal. So what we need to start off by bringing the blood supply in before it gets into the Haversian canal, and the way that we do that is through another canal called the perforating or the Volkmann's canal. So this is visible in this illustration here, but obviously we can't see that in a 2D section, in a 2D histological section.
Now that we've discussed compact bone, let's look at spongy bone. So as we move, probably in any long bone, as we move further away from the outside surface of the bone, so from here where the compact bone is necessary to protect the bone, and if we move closer into the middle or into the center of the bone, we get into an area of spongy bone that is intertwined with a lot of bone marrow, obviously housing red blood cells, stem cells, and other cells. So histologically, you can see that this image looks quite different to the one that we saw in the previous slide, and keep in mind that this is a low magnification. So there are still osteoblasts, osteoclasts, and osteocytes present here, but you can see there is a lot of marrow present around. What's the one thing that we don't see in the structure of spongy bone, Vidya?
So in spongy bone, we don't see the Haversian canals in here, and that's mainly because we don't need a system to try to get blood vessel into compact bone matrix, because it's a spongy matrix, and what that means is that it's a lattice-like network of scaffolding where we'll just have trabeculae shown in the histological images as dark pink tissue or matrix, and within that we have spaces or bone marrow where we find hemopoietic tissue such as bone cells, osteocytes, and obviously blood cells as well.
So this is a revision slide for you to use, with on the left those images labelled for you, and here on the right a great histological image that's labelled with all the cells that generally sit within bone. Vidya, looking at these 3 histological sections, I'm pretty certain that I'm somewhere within the female reproductive system. Can you help me out and identify exactly what I'm looking at over here?
Daniela, we're looking at the ovary.
Thank you, that's what I thought.
So the way that I've figured that out is that we've got lots of these ovarian follicles all around the top layer of the section. So as we're circling here, here, and here, all of these are individual ovarian follicles. They're not very developed at all. They're within the earlier stages, so they're called primordial follicles, but that's beyond what you need to know for your for the scope of this semester.
What are the cells that are labelled in arrows here?
So the arrows are pointing to cells that are known as follicular cells. So these are support cells that help the maturation process of these follicles.
So with that in mind, looking at the two histological sections on the right, even though the staining appears to be different, so especially with the bottom section here, that does not look like a H&E stain to me, but the structural feature to look for are the aggregations of these follicular aggregations that clearly indicate that we're looking at the ovary as opposed to any other organ of the female reproductive system. Vithya, seeing as you are the expert on the nervous system, can you please help us label this neuron that we're looking at over here? We've got some amazing images, histological images up the top, but just to remind ourselves of the structure of the neuron, go ahead.
So let's work off the illustration down the bottom and we can keep going back to the histological images for reference if need be. So we start off with these processes at the topmost image, and these are called dendrites. Now, dendrites are used by the neuron to receive information from surrounding neurons or surrounding tissue, following which we have the nucleus that's within the cell, or nucleus or the cell body within the neuron.
Is there another name for the cell body?
Yep, it's also called the soma, S-O-M-A. From there, unlike normal cells, the neuron cytoplasm just extends out to form the axon. All the way, yep.
And all the way here, yep.
Now, with some axons, what we can do, or specifically what happens, is we get myelin wrapping around certain regions of the axon. So in this case here, we're looking at a myelinated axon, and the myelin is reflected by these bluish coats that we can see around the axon itself. So that's why it's a myelinated axon.
So what about these gaps in the myelin sheath?
I'm glad you asked, because the gaps are really important for action potential transmission or neurotransmission to happen, because it propagates that nerve impulse really, really quickly, only because of these little gaps. Now, the specific name for these gaps are called, or is called, node of Ranvier. So finally, at the end of the axon, we have what's known as the axon terminal. And this is where we're expecting to see synapses, either with another neuron or with a neuromuscular junction.
So Vithya, is this a specific type of a neuron that we're looking at over here?
I'm glad you asked, Daniela. So it's a multipolar neuron. And the reason that that's important is because we do have other types of neurons as well.
Let's look at those in the next slide.
So in the far right image here, we can see the same multipolar neuron that we just labelled before. Examples or situations where we'll find these multipolar neurons are in the motor system as motor neurons. The reason they're called multipolar neurons is because we've got multiple dendritic branches, as we can see, inputting into that one cell body, and we've got one axon then coming out of that cell body. The other type of neuron that we have is called a unipolar neuron. Examples of these are sensory neurons, which sit within the dorsal root ganglion. And again, these neurons are called unipolar, pseudo-unipolar neurons, mainly because they have one branch of dendrites coming in with the information, and that information is then input into the cell body, for example, when it sits in the sensory dorsal root ganglion. From there, information is relayed into the axons. The final type of neurons that we have are called bipolar neurons, and these are often interneurons that are relaying information within other specific neurons. These are called bipolar mainly because they have two axons that we can see on both ends, one over here and one over here.
Looking at blood histologically, obviously what you'll be looking in a blood smear is for cells, because everything else around it is plasma and water and it won't be staining up.
So just to remind you guys, you did have an entire 1 hour review lecture on blood and hemopoiesis last semester. So go back to that lecture for more review slides if you do want that.
So let's just quickly label the cells that are in here, and then over the next 2 slides we'll talk about their function and structure.
So if we start with the most abundant cell type that we can see in this image here, these are all red blood cells. We know that they're red blood cells because they don't have a nucleus in them or any other organelles for that fact, and we'll talk a little bit more about them later. The next cell that we can see here is filled with really pinkish or eosinophilic or acidophilic granules, and that's because this is an eosinophil. Following that, we have a lymphocyte that we can see quite clearly. And the lymphocyte is identified by the fact that you have a nucleus occupying most of the cell. Towards the bottom here we have the basophil, and the basophil is the most purple looking or basophilic looking cell. More often than not, you won't be able to actually make out the nucleus because of the granules that are completely obstructing the nucleus. In the middle we have a monocyte, and these are the largest white blood cells which give rise to macrophages. And last but certainly not least, we have neutrophils, which are really, really important for us to help us combat bacterial infections and are important in a general inflammatory reaction as well. So I want to draw your attention to this diagram here, which we also went through in our last review session last semester. So it's a diagram showing the red blood cell maturation process. When the precursor cells, or as they're starting out, they have a really purple basophilic appearance, and as they're developing, they become more red or acidophilic. What I want you to take away from this is as they mature or as they become mature red blood cells, they lack or they lose their nuclei and also other organelles. So other organelles such as mitochondria are also lost in the process. Now, why would this be, Daniela? Why are they losing all organelles within the cell?
So the purpose of an erythrocyte, which we should all know, is to simply carry oxygen in their 120-day finite lifespan. The way they do this is by the carrying capacity of hemoglobin molecules that they contain and the iron present within it.
Excellent. So as they head towards the final stages of developing, just before they lose their nucleus, they're called just as soon as they lose the nucleus, they're called a reticulocyte. So don't worry about the term for the nucleus that's lost, that's not important. But as a reticulocyte, they do not have a nucleus, but they go into circulation, and a few days later, last minute modifications are made. They lose all the RNA, and that's when they become a complete erythrocyte.